it's an exciting time to talk to all of you. But Joni, your predecessor, Chris Austin, I know he's done it at this conference, and I've seen him do it elsewhere. But he used to run through a mathematical exercise on rare diseases, the number of the diseases, how long it would take to get therapies for each. But, if you wouldn't mind, can you just give a sense of that to the audience?
Yeah, of course. So many of you know, and in fact, we talked a little bit about this on the previous panel, but many of you know there are about 10,000 diseases. And I want to give a quick shout out while I'm doing that to Melissa Handel, who's sitting back there. Hi, Melissa. She's the one who actually came up with that number through a very deep analysis and looking across a variety of different databases to describe that number, because it's, in a way, we like to say it's over 10,000 because we really don't know the exact number. And the other thing that's really important to understand about that number is that when it comes to then translating that to the number of people, it's also a bit tricky. So over 10,000 rare diseases. Now in the US, we define rare diseases as a disease that's in fewer than 200,000 people. But that's not the same way that other countries define rare diseases. So a lot of what we'll talk about today in these numbers will relate to the US population. So 10,000 rare diseases. And you heard on the previous panel, if you were here, that it takes about 10 to 15 years to move a therapy to market. That's starting to shift, I think, given new modalities that we have out there. But if that's the sort of average that we'll build upon. And then we also heard the average of about $2.6 billion to get a therapy to market, accounting for a variety of failure and the processes for developing drugs. So 10,000 diseases, 10 to 15 years to make $2.6 billion. That's a lot of money and a lot of time. Now, if we were to develop therapies one disease at a time, 10 to 15 years per disease is, and over 10,000 diseases, that's tens of thousands of years. The math is really 100,000 years, but I'm going to say tens of thousands of years, assuming that there's going to be, some overlap there. And that is also, the cost of that is about 20 to 30 trillion dollars to do that. So we have to do something very different to move the field for therapeutics for rare diseases, or else there are going to be many rare diseases that are not even going to be seen. So if we think about the diseases, the 10,000 diseases sort of between my hands, we want to turn that so that they're vertical and that they're all sort of have the ability to come in and into the therapeutic development space in somewhat of an equal way. And so this idea of developing platform-based approaches is one of the key ways in which we like to think about that. So this more than one disease at a time approach is a critical feature, I think, that we have the tools to be able to bring to bear, to allow also industry, academia, government, patients and patient family and foundations and organizations to be a part of that process. So we're reducing the barriers to enter into the space of drug development for therapeutics. So I think that's one of the reasons why these numbers are so important to understand and to know in terms of how we approach rare diseases in general.
So about 20 years ago, we were all getting very excited about the advent of precision medicine and the idea that we are understanding the molecular mechanisms underlying diseases. And one of the consequences of that, though, is we've been defining diseases down. So, breast cancer became, not just HER2, but, HER2 negative, HER2 whatever. There's estrogen positive, whatever. But what's the consequence from a rare disease point of view of defining these diseases down? I mean, you just think about Duchenne, you have the exon skipping drugs, which work for a small percentage of patients. What does that do to the viability of drug development or the cost of developing therapies for all the people who need them? Any of you, all of you.
Well, I think maybe I'll start by just expanding on the math that Joni just outlined. So $30 trillion to correct these diseases over 100,000 years, right?
To warn people, Simon is an economist, so he
Yes,
does
economist and financial background, excuse me. So I think that, let's dig into those numbers. 30 trillion to treat these patients, right? If you look at the Every Life study, it costs the rare disease community $1 trillion per year. Using that math, it's 30 trillion plus 100,000 trillion in costs if we wait this long to treat our patients, right? I want to flip the question that you just asked on its head a little bit and say, what are the costs of not breaking down our diseases into their molecular bases? And cancer is a great example. Epilepsy is a great example in two different ways. Cancer has been breaking their diseases down into their molecular bases. There's no such thing as let's find a cure for cancer, right? It's let's find a cure for each cancer. And if I try to treat, melanoma or leukemia or glioblastoma the same way that I've treated papillary thyroid carcinoma, I'm going to kill patients, right? If I break it down to the molecular basis, I can start to identify commonalities. And if I identify those commonalities, I can start to build platform approaches around those commonalities. And we have so many opportunities to build those platform approaches within rare diseases, but without breaking it down and understanding what those components are that we can then dig into, develop. I want to define what I think platform approaches are: systematized, centralized, standardized, optimized processes that facilitate scale. The only way to do that is to bucket these things into logical categories of research.
Yeah, I would agree with that. The idea of breaking sort of people into lumpers and splitters. This is the idea of splitting, and this allows us to get at the molecular basis of disease. And I'll say that some of the approaches that we take to study this is to support a program that we call the Rare Disease Clinical Research Network. And within that Rare Disease Clinical Research Network, we support groups of scientists across the country who come together, but they have to study at least 3 rare diseases. And most of them study on average 9 rare diseases. And they're studying those 9 different diseases because they share some sort of symptomatology, or they may share some mechanism of action. But then that allows us to take a look across those different diseases, understand what's common across those, and then start to build the therapeutics to get at those more directly. They may not be the same therapies, but if we can understand the systems biology underlying them, then we can start to create the therapeutic approaches, the clinical trials, to bring in people with those very similar diseases in a way that we can study then different types of approaches for the therapeutics at the same time in a clinical trial. So it saves us on the development of the therapeutics. It saves us on understanding the phenotyping of the patients with the disease, and it also saves us in time in terms of developing the clinical trial, streamlining those clinical trial processes to understand how we can get to an answer faster. So I think those are this idea of precision medicine is really helpful. It sounds like it might take us longer because we are splitting, but in the long run, actually, I think it'll help us move as a field much faster.
And I think maybe for a lot of people in this room, many of the patient advocacy organizations represented here are focused on a pediatric neurologic disease. And such a great example when we think about how this has happened, because historically you would just have pediatric neurodevelopmental disorders. Maybe it would be called an encephalopathy. And now we know these very finite genes that cause them. But once you understand the gene and then the genetic mechanism, the biologic mechanism underlying that, then you start to find like your friends in the space, right? So you start to find like, okay, the gene that causes my child's condition is related to like histone acetylation, right? That this like epigenetic mechanism. What are the other disorders like that? Those are the ones we should be doing research with. Those are the ones we should be partnering with, because you can't all fund all the research needed yourselves, right? So you can find these associated disorders. There's been a lot of work done in ion channel disorders that are like this, where groups of these conditions come together and really fuel a lot of meaningful research.
So even though Simon is on the outside, he's actually sitting at the center of this panel in that he is both connected to NCATS and RareX. But Simon, I wonder if you might talk a little about your own experience. You had a daughter, Annabelle, born with a ultra rare neurological condition, and you set off to find a treatment. What did you go about doing?
So I think I'm probably going to describe half of this room's experience in rare diseases. So the very first thing that happened with Annabelle was that she was misdiagnosed with epilepsy when she was 2 months old. And if for most of you, you probably know this, but to get diagnosed with epilepsy, all you have to have is 2 unexplained seizures. That's it. You're in a big bucket with a whole lot of other people. And they give you Keppra and trileptal and valproic acid, and they tell you to go and take it and see what happens. That was our lives for the first 2 years until she got a genetic diagnosis. And when she got the genetic diagnosis, it was alternating hemiplegia of childhood, no epileptiform seizures. She was taken off the drugs that she was given. And that started us on the path of research, right? So the genetic molecular basis of the disease started our path. And that was really the catalyst. But what it did was it frightened the hell out of me, because now my best friend was dying and I had to figure out what to do about it. So woke up at 3 a.m. every day. This is not an exaggeration. Read as many scientific papers as I could on AHC and what had been done in it, and started to categorize those research papers. I would read those all day long until I was exhausted, fall asleep, and wake up at 3 a.m. the next day. The first paper took me 16 hours because it was all Greek, and I really wanted to understand every word in every paper. I read 400 papers at least and categorized them, and then hired somebody who worked at the NIH, Dr. Natalia Morsch, who's just fantastic background in molecular and cellular biology, to help me to figure things out and to categorize. And we categorized those research topics into various different categories. The first was structural defects. The second was function and functional consequences, which led to pathways and other genes. Tools like animal models and cell lines and assays and biobanks, and these types of tools that may be available to us that we needed to develop. Therapeutic options, things that have worked for other similar diseases, and then how we might be able to develop platform approaches. So that was the path, but really the genesis of the ability to create therapies came from the molecular basis and the genetic diagnosis.
One of the things you saw, though, in learning about this disease was its commonalities with others. How did that affect your long-term thinking?
So that's, and I talked about platform approaches being systematization, centralization, standardization, and optimization. I've walked the path of many others, right? I'm not a trailblazer. Everything that I've done in rare diseases is following somebody else's path. Now, it might be following the path of, fighting my way through weeds, or it might be following a super highway that someone's created that, I can just walk down or drive down in a race car. But in each one of those cases, I've been reinventing the wheel, right? And in each one of those cases, there are commonalities. So when you look at the opportunities for platform approaches, there are basically 4. So data, I think, and RareX is doing this fantastically well. Centralizing, systematizing data so that it's usable. Tools, mouse model, cell lines, all these things. Therapies, whether it's AAV-mediated gene therapies, ASOs, RNA options, gene editing. These are all paths that all of us can explore. Maybe not take, but explore, right? And then there's that systematized, categorized research that is the fourth of these things. If we do these 4 things in a centralized, systematized way, according to a strategic plan, I feel like we can really make advancements. So that was the path.
Joni, you've been involved in actually building a platform approach, thinking of the bespoke gene therapy consortium as well as the Ph.D. program. Can you explain what those are?
Sure. I think you've articulated sort of the concept that, 10,000 diseases, but really there are many fewer mechanisms that cause those diseases. So we talked about AAV mediated to correct a dysfunctional gene, or the tRNA therapeutics that you mentioned earlier, Danny. The Bespoke Gene Therapy Consortium and the Platform Vector Gene Therapy Program, or PAVE-GT, these two programs are essentially to describe how we can systematize, I'm not going to remember all four, systematized, centralized,
Standardized, optimized,
and optimize these approaches. So the platform model, what he described is spot on. The idea is with Bespoke Gene Therapy Consortium, as an example, we're working as a public-private partnership with industry partners, with biotechs, with patient organizations, and with federal partners at NIH, as well as at the FDA. And this is all coordinated by the foundation of the NIH to help align those programs and projects that are within BGTC. So the idea is that we would be selecting diseases that would be amenable to AAV-mediated gene therapies. And we selected 8 different diseases through a very sort of rigorous process of identifying these particular diseases based on a criteria of a patient population that was available, the sort of the amount of time that would be needed to assess whether it would be working or not, and then also that there couldn't be any commercialization potential for those diseases. There couldn't be another company working on them. We don't want to compete with companies. And so those were the criteria that we used to bring the diseases that we're exploring in BGTC in. And then the idea was we wanted to select diseases of different sort of targets. So we have 3 eye diseases. We have 3 neurological diseases, and we have 2 liver diseases. So 3 very different organ systems. But the point there is really to understand how to use the AAV-mediated approaches as a platform across those different diseases. And so, for example, in the eye diseases, we're using a particular AAV vector. There are about 12 different AAV vectors that target very specific tissues. So the vector that you choose matters. It goes to a very specific site. And those AAV, it's a virus, but it's a benign virus. So when it targets those cells, it does what viruses do well. They deliver the content into those cells. And that content happens to be a functional copy of the gene that can replace the deficient copy that the patient might have. So if we can understand how to build the platform of packaging those different genes that cause those 3 different diseases in the eye, we want to make 3 different packages, but we want to use one single AAV. And that then becomes a platform. So we can then define and systematize that single AAV shell or package, capsid, I should say. And then the cargo that goes into that, the gene, the therapeutic gene, that's the key difference. But what we want to do is make sure that we can describe that the AAV that we use to deliver the cargo, we do all of the chemistry, the manufacturing, the control work, the regulatory science work to ensure that it's made in a consistent fashion and safe to deliver to patients. But then when we change out the cargo, that's the only thing that the FDA would then need to review. So it minimizes the work that the FDA has to do if we're able to create that sort of a platform-based approach. So it reduces the sort of one-off nature that we have of gene therapy in this space and puts it really more into a platform kind of an approach to it. And it also reduces the requirement for the FDA to review a lot more material. But when we can we can essentially pull an AAV, caps it off the shelf, and put just a new cargo in. They don't have to worry about that piece. They only have to worry about the new cargo. So it really capitalizes on this idea of a platform, and it again reduces that entry for the barrier to entry for people to then figure out how they might be able to use that platform for their work or their disease. So our goal with the BGTC program is to show that proof of principle and then create that as a publicly available resource for others to follow suit, and use that playbook, if you will, for advancing other diseases too.
One of the encouraging things about this, I think, is that Peter Marks of FDA is co-leading this. How much time and cost might be shaved off of developing a gene therapy if you're able to just swap out that gene for the other one that's been through the ropes?
Yeah, I think the cost in goods and manufacturing, all of those things, we still don't have a good sense of at the moment, because the manufacturing aspects, as it scales, the manufacturing is going to need to scale with it. And right now we don't necessarily have a good path for that. So I think that question about the actual costs, savings that we will see, remains to be seen. But the cost in savings of time and resources for the platform-based approaches, I think that's very clear. There's so many things that go into the cost of a particular medication or a drug. You've heard the $4 million price tags of gene therapy. Will it reduce those costs? That also remains to be seen, because it's not just necessarily how you make the drug that goes into developing what a cost price point would be. So the idea, though, is that if you can reduce those costs, that should factor into then what that would look like.
You threw out the term playbook a minute ago, but you meant that quite literally. There is a playbook.
I
So
did.
what value might that have to people in the audience?
Yeah.
Find it.
This playbook is something I think if you're in the space of gene therapy or even gene editing or ASOs, biologics in general, it's a I think very informative playbook for people who may not necessarily be in the space. And what you can do is I don't have a QR code, but you can Google BGTC and playbook, and you will find in the websites then the playbook. It's about 80 pages, and it really goes through the process of how gene therapies are developed, and what it takes to do the manufacturing, the chemistry, manufacturing controls, the regulatory processes of developing an IND package, the regulatory processes or the incentives of rare pediatric drug designations and orphan drug designation packages. Those are all templates in the playbook that you can take advantage of and apply them to your own disease as well. But it's a nice compendium too that talks about how to work with the FDA. When should I meet with the FDA? What does that meeting look like with the FDA? What kinds of questions should I ask them? There are literally checklists within the playbook that will help guide you in exactly the process for doing that. The playbook is going according to our playbook and in the BGTC program. We're entering our third year, ending our second year. And that playbook is in that first stages of development. So more of the regulatory transition from the new investigational new drug approvals, IND approvals, that process, the regulatory processes for manufacturing, into first in human clinical trials. But as we proceed through the BGTC program, we will start to update our playbook with more experience that we have in the program as we get more into the clinical spaces too. So we'll talk a little bit more about patient recruitment, protocol development, et cetera. So it's something that we're expecting to iterate over time, and I would encourage you to continue to go back to it and take a look as we do update it in the next few years.
So BGTC is about gene therapies, but I'm wondering, any genetic medicine I think of as being a platform, at least that, if you're dealing with an ASO or you're dealing with other RNA therapies, if you're dealing with gene editors, there's a potential to replicate that kind of an effort. Are you thinking in terms of that at all?
We are, great questions. We are definitely thinking about this as well, that we have a big program in gene editing called the Somatic Cell Gene Editing Program, and it's a very similar process. When we started both programs, we thought that gene editing was going to be further behind, but it is moving rapidly in this field, and there have been great strides made. But the idea with BGTC is, you're right, Danny, it's sort of a platform of platforms, if you will. And so what we're learning in the gene therapy space of AAVs is transferable. Not everything is going to be transferable, mind you, but there are going to be some areas that will be transferable. The lessons learned will be transferable. The working with the FDA will be transferable. But the specific changes in the modality then could be highlighted. But essentially, that platform should be transferable to essentially a gene editing platform or an ASO platform or other kinds of new modalities that might come along. So that is the hope with building this platform. It's meant to be sort of a way to help create for the other modalities out there too.
So one of the things we've seen primarily in cancer, but not limited to cancer, is basket trials. I know NCATS has been involved in a pilot program on basket trials. Can you explain what a basket trial is and what that might do to advancing therapies for rare disease?
Yeah, so the basket trials are essentially a type of a clinical trial. And this is, you mentioned cancer earlier too. You don't treat cancers, you treat the specific cancer. And so in the field of cancer, we learned that there might be very different cancers. So how can we think about bringing in the very different cancers that might be treated by one particular drug? And so the cancer field, we learned from that, and we said, hey, how can we apply that for rare diseases? Because we know that, for example, there are a variety of rare diseases, and I'll just pick on the gene therapy here too. This is really the whole premise behind the Platform Vector Gene Therapy Program, or PVGT. And essentially it is bringing in looking at two different diseases that are caused by 4 different genes. And so we were creating a gene therapy for one gene. For the propionic acidemia is the example here. One gene causes propionic acidemia. Another gene, different but in the same in the same sort of biological cycle, the system's biology. It has a different gene, but it causes the same disease. So here again, all we're doing is replacing the gene in the same exact AAV vector. And for the platform approach, we can then bring people who have actually different diseases. They're caused by different genes, so they have different diseases, but we're putting them into the same clinical trial. So we can use maybe one control arm, but 2 different groups of patients who will receive 2 different genes that will be therapeutic for them, and study them at the same time. So here that's what a basket trial is. Essentially, you're looking at different diseases, but you're treating them in using very similar kinds of approaches. In the gene therapy scenario, the adeno-associated viral mediated vector delivery is what we're testing as the same thing across those different diseases. And so this helps us understand whether or not that is, first of all, going to work. We think it's going to work, but we don't know until we test it. So that's why we have to do the clinical trials. But what it does for the FDA and why they're so involved with this program is then it helps them make decisions as to whether or not how they start to review packages that come afterwards, and can streamline their processes for how they do that review. So that's where the basket trials really come in to be create an efficiency around looking at various different diseases, but that are being treated with the same kind of modality. So that's the real value and benefit of those.
Simon, you talked about the fact that your daughter has a rare epilepsy. Now, it seemed to me that this is a perfect case where a basket trial might be compelling. Are you seeing any activity like that, or are people hesitant to pursue those kinds of approaches?
Yeah, so I'm seeing real opportunities. Now, I did describe how, layman, economist, finance guys are kind of being a hub for rare disease research, and we've seen that in so many ways. I think it's still kind of in the nascent phase for my disease right now, but there are huge opportunities, and I'll describe them a little bit. So in our disease, it's associated with neurons. So my daughter has a defect in what's called the sodium-potassium pump, which allows neurons to fire and keep firing, essentially. There are many other genes associated with that pathway, right? So for instance, you've got voltage-gated sodium channels, voltage-gated potassium channels, and calcium channels that allow for neurotransmission and for neurons to fire and keep firing. If you look across those genes, you've got patients with the same disease, different genes like we were describing. So there are real opportunities to work together on various platform approaches, whether it's centralized data, centralized research repositories, centralized cell lines and animal models, and centralized therapies. If we look at the playbooks, we look at, how do we take something that's worked very well, optimize it, and then create a superhighway for others to follow across linked diseases, whether it's linked in terms of phenotypes or linked in terms of, genotypes. Let's do that. And we're starting to have conversations now with genes like SCN2A, it's a voltage-gated sodium channel, and CACNA1A, which is a calcium channel, all along the same pathway to try to centralize that research and save time and money to be able to get to the finish line using these systematized, centralized, standardized, optimized approaches.
We need T-shirts.
We need to acronym.
So obviously there's been a lot more experience with basket trials in cancer, but are there unique challenges to doing that in rare disease?
Oh, yeah, I think there are. I mean, largely the challenges were described also in the previous session, sort of just the numbers of patients. I think that's one of the challenges. But I think, the whole goal of projects that we in the federal government can support, the idea is to take on the risk of testing these ideas, because if they work, they can become super highways. And so I think part of it is understanding how to take on that risk so that others don't need to do that. And that helps us then get to that efficiency faster. So I think there are challenges, though, not just in sort of the patient populations, the small numbers of patients, but also in, again, this is a new area that we are not scaled up and ready to go. So manufacturing is going to be a key space for this. But I think these are challenges that are almost engineering challenges that we need to work through. But there are also economic challenges. What are the economic incentives to make sure that we as a nation think about prioritizing these kinds of approaches so that they can be subsidized in how we approach the manufacturing and the capabilities to develop them further. If it's okay, I wanted to also pick up on something else that Simon said in the idea of the model usage too. A part of the Ph.D. program is we've also been developing cell-based models that are from rare disease patients. And I talked about propionic acidemia as one of the arms of Ph.D. The other arm is actually myasthenia syndrome. So these are neuromuscular junction diseases. And so what we've done there is we've taken cells from patients who have the disease, and we have created basically tissues from those cells. And we've put those tissues on a plate where we can look at the functional readout of whether or not that muscle twitches. And so it's a pretty sophisticated model, but it's actually pretty easy to build. And it has a really nice functional readout that we can test these gene therapies in the model before they ever go into patients. And so I think also on the preclinical side, making sure that we have the platform approaches to develop these models too, can help also streamline the pathways to IND approval and then moving into patients with a higher confidence.
So Karmen, I wanted to talk to you about the way RareX is enabling researchers to look across diseases, but it dawned on me that in the audience may not really be familiar with RareX. So maybe if you wouldn't mind explaining what the RareX platform is.
Yep, sure. So RareX is a platform that was developed really in response to a need from the community. So you've all probably heard in the last day and a half, at least 15 times, get data, collect data, get out there and make sure you know who you people are and get them to provide data. And the feedback that Global Genes received a number of years ago over and over again was, well, we're trying to collect data, but either there's our academic partner who's great, well, holds on to all the data and they're not sharing it with others. And we have other researchers who really want to access the data and can't. Or we went out and got quotes for how to build our own really, very customized database. And it's crazy. It's hundreds of thousands of dollars and we can't afford that. And if we could afford it, that's not what we want to put that money into. So RareX was created really to be this place to for patient advocacy organizations to come in and to have a partner in us to be able to collect data and to collect it in a way that is very systematic. It is robust. It is research grade data. Now, if you're going to collect research grade data that is really genuinely going to be useful to propel research forward, you have to be very, very thoughtful about how you collect that you ensure that the way it's collected is in a systematic way that can be compared to other data sets. And doing that one disease by one disease is completely not feasible, right? So the way that RareX collects this data, the way that we sort of develop how we collect the data is we look at groups of disorders. So RareX got started in neurodevelopmental disorders and has expanded into about 5 other disease areas beyond that large umbrella. But if we take that example of neurodevelopmental disorders, we convened a large expert working group composed of clinicians and industry researchers and patient advocates to come together and say, what are the most significant symptoms and impacts to daily life that we see across all of these disorders? And how do we just systematically start to sort of attack those? Because we're not going to add them all to the platform tomorrow. Let's go out and let's identify and then choose what's called clinical outcome assessment measures. So these are essentially surveys that all of you have experience doing in clinic that you have usually no idea how much research goes into. Like these surveys that seem quite simple, there's been a ton of research to understand, is this really what's called fit for purpose from the FDA? So is it really capturing what's most important to this disease community? And does it capture it in a way that is quantitative? Because if we're going to collect this data and we're going to hopefully use it in a clinical trial, we have to ensure that there's room for movement, that there's a quantitative score that comes out of it, and that there's opportunity to see movement. If everybody in the entire disease community falls to what's often called the floor, everybody has this low level of a score, that's not useful. We need to see a range of scores. So we do all this work together, and then we license and implement those measures onto the platform. And then it is a virtual platform that is essentially an opportunity for patient advocacy organizations globally to then invite individuals in their community to participate. So is it as robust as a natural history study at a clinical trial site? Nope, right? We're not going to be able to collect all that clinical data. But what we've seen in many groups is that we sometimes are able to engage 3, 4, 5, and more times as many people as exist in any of the current natural history studies, because we reach people where they are, whether that's a rural area or somewhere globally.
I don't think you said this, but just for the audience, who pays for it and who owns the data?
Oh, yeah, that's important. So Global Genes is, I think, as all of you know, a nonprofit organization, and RareX is the research arm of Global Genes. So it's nonprofit funding, which is not easy. It is not easy. But we get that through a variety of grants and sponsored projects, that funding. And
Free to
patients,
patients.
it is free to patient advocacy organizations. So we have a base platform that every patient advocacy organization who joins and who comes on board and starts to partner with us gets all that free. We do have some larger research consortia programs where we build on top of that, where we do get sponsorship dollars, and sometimes some of that comes from the advocacy organization and sometimes it comes from biopharma. But importantly, I don't want to forget to answer your other question, the data itself is owned by the patients. So our commitment is that this data is and always should be available to a wide number of researchers according to patient preference.
So I know you did the challenge last year, the data challenge, but are you at a point now that there's enough data that researchers are using it to look across diseases, and what's the potential for that platform to help people see connections across diseases?
Yeah, well, I think, it's interesting. You've heard a lot here today about thinking about mechanisms, right? So Simon was talking about these ion channel disorders, whether it's a sodium channel or a potassium channel, and, really thinking about what's the underlying cause of the disease. Sometimes it's useful to group disorders not by underlying disease biology, but by symptoms. What are the shared symptoms? And so that we do a lot of that on our platform, and thinking about what are the symptoms that are of greatest impact to this patient community, and making sure that those get prioritized for what patients and families are asked to contribute data on. So if it's a condition where a severe communication deficit is the single number one most important aspect of the disease that families report, like, I really struggle to communicate with my child. That's the number one thing that we would like to see changed. I want to be able to communicate with my child better. Then we need to make sure we're capturing that really well. And so currently, every single parent of a child who has a neurodevelopmental disorder with a severe communication deficit gets the very same standardized survey. And so we do have some researchers really interested. And in fact, we're embarking on a collaboration with Duke that developed that measure to look at sort of platform ways to work with the FDA to validate that across a lot of conditions at once, instead of really the very long process that traditionally exists.
You also started consortiums. In May, you launched the Sleep Consortium. What is the Sleep Consortium, and does that allow you to look at symptoms across conditions?
Yeah, so I mentioned that we do have opportunities for these sort of like plus projects, right? There's our base platform, and then there's some of these larger research projects. So the Sleep Consortium is a good example. We currently have 3 active, large, multi-stakeholder research consortia programs. The Sleep Consortium is interesting, I think, because it's primarily focused on rare disorders of hypersomnia. So think about narcolepsy as a very classic example. These are extraordinarily difficult disorders to diagnose. You wouldn't think it. Doesn't seem like it would be, but they actually really are. And throughout the world, and even within this country, the way that different centers do polysomnograph testing, do other types of sleep testing varies widely. And it can be extremely difficult to diagnose these conditions. And yet, and this will resonate with a lot of people in here, you have these estimates that it's so much more prevalent than what they can find, who they can find in their population. And so if we step back for a minute, we just say, like, okay, who has a sleep-related disorder? Who finds that sleep is a massive problem? And then within that, you start to bucket it down into sort of too much sleep, too little sleep first, right? And then you get into deeper symptoms, and then you start to get into the underlying diagnoses and reasons. And so for them, for those patient advocacy organizations that are participating in the Sleep Consortium, part of the reasoning behind it is collecting this robust data for research. Part of the reason is really identifying patients through this sort of just symptomatic self-report mechanism.
So each of you are trying to pioneer, excuse me, new ways for people to pursue therapies for rare diseases. I'm wondering where you see the biggest challenges. Are they financial, regulatory, scientific? Like all of you to weigh in.
I'll take a shot. So I think that in a lot of these cases, we've solved the problem, but we've solved it at a silo level. So, and I'll talk about opportunities first, just building on what Karmen just said, because I think it's so important. We've got 10,000 rare diseases, and if you break it down to the molecular basis, we've got multiples of that. We have such an opportunity. In business, you look at your strategic plan, you look at what, optimizations or efficiencies you can create, and you identify what the lowest hanging fruit is, right? I think in rare diseases, we've got one obvious set of low hanging fruit, and that is data, right? Gathering and centralizing genotype and phenotype data. And Karmen has just explained what we can do with phenotypic data according to domains across multiple diseases. Imagine a world where we've got all of these markers, all of these diseases, all of these molecular bases of diseases with their genotype and their phenotype associated mapped. That's a Rosetta Stone of human biology, a Rosetta Stone of human function and dysfunction, right? We have that available to us as a society to go out and get that, right? Genotype and phenotype data should be centralized, should be a public good, should be accessible, right? And I think we're getting there, but we're nowhere near it right now. So I think that's one real opportunity for us. And then I think the very first question that someone will throw back at me on that is, how do you de-identify? Well, that's something that I think we've solved too, right? I can't see your bank account, Danny, right? And I haven't been able to for millennia. We've solved that problem, right? We've got personal identification technology that can blockchain that can prevent us from accessing who that underlying person is, but the data is valuable and rich and gives society the ability to really create an underlying database of tools and knowledge that we can use not only to create therapies and tools around, but to really understand ourselves as humans.
Is the United States lacking here? I mean, is the UK ahead of us in terms of doing that? Are other countries ahead of us in terms of doing that kind of central data repository?
Yeah, so I think I'll give one example. So you should all look this up, this particular paper. It's called Diagnosis of Rare Disease Pediatric Patients in the UK and Ireland. It took 13,500 clinically undiagnosable patients, right? Clinically undiagnosable. And it ran a whole exome sequence, not even a whole genome sequence, on those 13,500 patients. And then it ran multiple regression analysis, which if you go to a statistician, it's like trying to kill a woolly mammoth with a cucumber, right? We've got precision guided missiles, but we're chasing a woolly mammoth with a cucumber. And of those 13,500 patients, we diagnosed 5,000 immediately, right, with a disease based on their exome. And another 3,000 of those patients, we got disease associated, like strongly disease associated variants of uncertain significance. So that's 8,500 patients out of 13,500 that now have direction, the ability to go out and do proper research. So yeah, I think they're a little bit ahead of us in some cases, right?
Yeah, I think that's accurate. And part of the issue is that we're very fractionated here in this country in terms of the health systems and how you get a coordinated effort like that underway. And, I think there are ways in which we can incentivize that. But I liked how the financial guy talked about data and science in his answer to your question. And I'm going to be the science gal that's going to talk about the financial issues. But I do think that this idea of incentivizing the study of rare diseases, the treatments for rare diseases is still a big challenge. The commercialization potential is something that we also need to think about as a platform. What are the areas in the commercialization space that need us to help that incentive to get that as more of a platform-based approach. And I think one of the ways that we think about it is if you're in industry or biotech, you're thinking about the return of interest on the work that you do. What are you going to get back so that you can support other work that you do? In the federal government, we don't get money in return. We are evergreen based on the congressional appropriations that we get through taxpayer dollars. And so we have the opportunity to use those for sort of this idea of a greater ability to address bigger problems. And in that way, our return of investment is, our approach to that is really about de-risking the entire translational science pipeline. So the more that we can de-risk it so that we can platformize these kinds of approaches, democratize the ways of getting academia and others involved in the programs and patient groups and otherwise, that is one way in which we can de-risk those the space, so that then perhaps if we de-risk it to a point, then all of a sudden it becomes commercializable. And so then we have to figure out how to incentivize companies to take those on from there and move them forward into that commercialization space and get them to the patients that need them.
We're going to open this up for questions from the audience. If anyone has a question, you're welcome to raise a hand.
So I'm actually going to start off with the questions that we've received in the app. And the first one is for you, Dr. Rutter. Could a basket trial work in a single disease state testing generic drugs for off-label use?
That's a question?
That is a question?
Yeah.
Would you like
it's not limited to any generic or otherwise. It can be used for any type of drug. Generally speaking, and this is another area of incentivization, but generic drugs are there's not a lot of commercial gain from studying generic drugs. There's no money to be made in that. So generally speaking, that's not a path that's going to be as fruitful. So the platform basket trials are generally used for a variety of different drugs. They could be used for generics too, but it's a way of really thinking about different diseases that can be treated by one path.
Thank you. The next question is for Karmen. How does RareX address patient priority? Do you offer surveys that ask about priority?
That ask about priority?
Priority. I believe like priority for patients and caregivers.
So this is something that we undertook in a limited way last year. If you think about the way that I described the platform and these existing standardized surveys, what happens is when you look at the data that we have on the back end, we have data on what are the most common symptoms in a particular disorder, right? So in any one disorder, if you start to look at the data, we can see what are the most common symptoms reported. If you look deeper into the data, you can see how that affects certain aspects of quality of life or activities of daily life or like work, school, absenteeism, things like that. But you don't get what Kelly just asked. You don't get, if I were to rank all of the symptoms that my child has, here are the top 2 or 3 that impact her life the most. And by the way, also please ask me, because I'll tell you that like the 3 that impact my life are probably a little bit different, but also important, right? Like she doesn't care if she doesn't sleep through the night, but it's killing me. So we undertook a project last year, and CACNA1A was one of the primary motivators of this, helping us to think about like, this is kind of a custom question, and yet it's a universal question. Everybody needs to know what are the most burdensome aspects of the disease, both because in the way that you think about sort of approaching the entire community, that's important. But if, we're all here talking about therapeutic development. If you have therapy developers who are focused on your disease, who are developing treatments for what's number 5 on the list, guess what? Not only is that not super impactful for your community as a whole, but you're going to really struggle to enroll a trial. You develop a therapy for the thing that 80% of the population says is their number 1 most burdensome symptom, and it's going to be a whole lot easier to enroll that trial. So this is not currently available across the entire platform because it takes a fair amount of work on the back end for us. So we look at all the data to see what are the most common symptoms reported by the community at large. We don't want to go to the existing literature. In very rare diseases, that might be 15 patients. That is not representative. You know, if we've got 60 on our platform, we want to go to that data instead. If we've got 200 on our platform, we want to go to that data instead. And then we work with the patient community to understand, what are people telling you? What do they tell you they struggle with the most? And then we merge those together to come up with the survey. So it's something that we're looking at in next year to really like make more widely available. And it's something that I think some of the industry partners we've been talking to are willing to sponsor and support.
Thank you so much. We are actually running pretty short on time, so I'm going to hand this back to you to close off.
Let me ask you this. I think one of the things that people in the rare disease community that are looking for therapy, they often feel such a sense of urgency that it's often dealing with diseases that are progressive, and it takes time to get to a point, even if you can dose them in an experimental therapy. Is there an advantage for them to be looking across diseases? Will that help them get there any faster, or is that more of a broader scientific approach where, you know, disease communities that are still going to be focused on getting their fastest path, you know, taking a well-trodden path?
Yeah, I think that's absolutely an approach. I think if you're working in a disease that there are similar symptomatology, for example, or other genes within the pathway, like the ion channels, I think these are areas where you would definitely want to consider working with others, learn from what they've done as well, and bring them into the fold, so to speak, find your people, if you will. But that sense of urgency is definitely felt and very important. And so in addition to the sense of urgency, there's also this issue around setting expectations, because the diagnosis is one piece, and often it takes on average 7 years to get diagnosed with a rare disease. We're working very hard to shrink that down, but that window of 7 years on average is a long time. And so you can age out of the modulating therapies, for example, and those diseases can become more severe. So that time component for diagnostics is incredibly important. But then once you're diagnosed, you have this treatment odyssey. And that's, I think, where leveraging the work we talked about splitting, but then lumping initially to find others in that space. I think you have a lot to learn, and so it can help with that urgency in moving more quickly in the space.
So since you lobbed it up, I'll let people know that at 2:00 in this room, we're going to be talking about using AI to shorten or eliminate the diagnostic odyssey. But you wanted to
Yeah, I wanted to follow up on what Joni just said. I actually really like the idea of diagnostic odyssey, research odyssey, treatment odyssey. But we started off this panel by talking about math. And the basic math is, right, if my child is dying, it's urgent, obviously, right? But 6.3 years, 7 years to diagnose a patient from symptom onset, not good enough, right? The 100,000 years to figure out a treatment for all of our rare diseases, obviously not good enough, right? It becomes a necessity. It's not about, should we be thinking about multiple diseases at one time with a platform approach? We must. I mean, that's the only way that we reduce these timelines. That's the only way that if you figure out today that your child has a rare disease, you're ever going to get to the finish line within, you know, to be able to save that child.
So we've got about 2 minutes left, but I've just asked each of you to offer one parting thought that people can leave here with and actually take action on. Is there any advice you would offer?
Find your partners, find your community, find your partners, and learn from each other. Stand on the shoulders of people who've come before. I think that's one of the key things. I think we've talked a lot about data and the importance of that, sharing those data and working together. Finding those partnerships and the people that you can work with is really important. And so that's what I would offer.
I'd say walk away with commonalities. We're all basically commoditized widgets. We're 99.6% genetically identical. We've got all of these ways that we can identify ways to work together and, collaborate to be able to have a really powerful set of people, 10% of the world's population. Let's go out there and gather, centralize, systematize, standardize.
T-shirts.
Right before we came up here, somebody said to me, I think in each of these sessions, I just need one thing, one takeaway. And so maybe it's perfect that I think in some ways we're all three saying the same thing. Like, you are not alone, right? There are people in this room, there are people at this conference, there are people more widely who are, if not the same disease, it's the same group of diseases, it's the same kind of symptoms, and it's the same journey. So there's no reason, to try to do it alone.
And if you need shoulders, there are some big ones here. You can just take your shoes off so you don't get stuff. But please join me in thanking this great panel.